Then now you are done with the electrophysiological recording. Then we are going to stain our preps with two new dyes. You already stained it with methylene blue, but this time we use two new dyes. They are 2-di-asp and also the cobalt chloride. Then for 2-di-asp, this one is a vital stain. We use 5 micromolar or for 2-di-asp vital stain. Then this one will stain the preps, and the prep will stay alive. It doesn't kill the prep. And also we can look at it under the fluorescent microscope. Also we should remove most of the preps, because when you are done with the electrophysiology, we don't need all the segments here. We should cut the prep through carpus and proper joint, and also cut the dactyls too.
The goal of this part of the dissection is to obtain the longest possible segment of PD nerve. You'll separate the PD nerve from the main leg nerve with a glass needle, and then sever the PD nerve as close to the main leg as possible.
And also we can get the tension, like the PD organ nerve here. Then we can see that the PD organ nerve this is the PD organ nerve. And also we have the tendon of the closer muscle. These are closer muscle fibers, and also a part of the dactylar segment. Then we should make a well and put this nerve inside the well, and put the 4-di-asp dye inside that well, and it goes through the nerves, and also it will stain the PD organ nerves. Then now I'm going to make a plastic platform using a weight boat here. Then I will cut the plastic from here and like a V shape, and here you see that it has a smooth edge. Then when we put the nerve over it, because we have to make a Vaseline well here, and when you put the nerve inside the Vaseline well, then it won't pinch the nerve, because if you pinch the nerve, then the somas won't stain very well. Then I will cut it here. Because if you put it inside the saline, you see it's going to float. Then therefore I'm going to make two holes here with a pin. We can make two holes here with a pin, one here and another one here. You see that it has two holes. And also we can use small insect pins to fix it inside the saline. Then it won't float inside the saline. Then now I put two small pins inside the holes that I made before. Then now when we fixed the plastic platform with the two insect pins, then we should raise it a little bit. Then it should be a little bit high. Then we have PD organ here, then it should be higher than the PD organ. Then after that we should remove some of the saline, because the plastic platform should be dry. Then now I am going to rub it across the surface, but we still see some saline there. Then therefore I'm going to remove more saline now. But make sure that the nerve is not close to the tissue, because it makes it twists the nerve. Then now I'm going to make multiple layers of Vaseline here around the well. Also, you can push it with the needle. Make sure that it's closed up very well. Then after that, now we have a nice well. Then I will grab the nerve. Then now I am going to add some saline into the well, because if it's dry, then the nerve will stick to it, to the plastic. Maybe just a drop with micropipette here. Then also I'm going to make a V. Then I'll put the nerve through it like this. But then I'll grab the nerve and put it inside the well. Then you can grab it at the tip, then we can cut the tip later on. Then now if it's too long, just let it hang over inside the saline inside the well. Then we can see the nerve inside the Vaseline. And now I'm going to use a tissue. Then you can make this and also put it inside the well. And after that, you can see, if you look at it under the scope, then you can see it sucks up all the saline, because we should put the distilled water inside instead of saline. Then when you add distilled water, then if you see that it drips, the distilled water doesn't stay inside the well, it means that it's leaking. Then we should fix it up before we add dye. Now I'm going to remove the distilled water inside the well. Then now you can use this little wick. Then it sucks up all the distilled water. Then after that, I'm going to add 4,2-diacid dye. Then you see, I will fill it with this. Then now I'm going to add more Vaseline around the well and just make a circles, because when you leave it like this inside the fridge or here, then it dries out, the 4,2-diacid solution will evaporate. In order to prevent this, then I will make circles with Vaseline around the well until it makes like a dome. Then it covers up the whole thing, then it prevents evaporation. Then if you remember, like, we removed the saline, and now I'm going to add some saline to keep the prep moist. But be careful, don't flood the well with the saline. You should be careful here. We want to keep the nerve moist. Just make sure that the nerve is inside the saline. Yeah, now it's inside the saline. Then also we should cover the prep with 4,2-diacid with something like this, with the cover, and put the cover on it. And you should keep the light off it, because it's 4,2-diacid and it's very sensitive to light. It will degrade if there is a very intensive light on it. Then after, when you leave the probe inside with the nerve inside the 4-2 diacid dye for 1 or 2 hours, then we can remove the dome that we made above the well. Then carefully remove the dome by tweezers. Then after that, we can take out the nerve and we can look at it under the fluorescent microscope to see the neurons in the PD organ. And also we can wash the well to remove the 4,2 diacid. And also, if you look at it under the fluorescent microscope like this, without removing the 4-di-2 aspect, then you see that it will be very bright and you cannot see the neurons. But we have to be very careful when you use cobalt chloride instead of 4,2 diacid dye, because the cobalt chloride, if it spills into the dish, then it makes all the dish black. Then we don't see anything. Therefore, in case of cobalt chloride, it's better to just cut the nerve. The nerve goes into the well, and don't remove the dome of the well. Just cut the nerve. Then we can remove the whole prep into another dish, and also we can do the precipitation of the cobalt chloride. Then we can take our prep into the hood, because cobalt chloride is toxic, and also we have to use ammonium sulfide to that ammonium sulfide reacts with cobalt chloride, and it will precipitate the cobalt chloride, and we will see the black stains inside the neurons. Then after, when we incubate our prep inside the cobalt chloride overnight, we put it inside the refrigerator. It should be cold, and also you have to be very careful because the saline evaporates, and we should add more saline and keeps our prep moist. Also, we should put some tissue around. Here we put some tissue around the dish that contains the prep. Then it makes, or the prep in this case stays moist. But also we have to be very careful and add more saline, like after like 6 hours or 8 hours. Then also we should work. Then after that, when we are done with the incubation, we take out the prep and put it inside the hood. We should work inside the hood because we deal with the ammonium sulfide. The ammonium sulfide is toxic, and we should work inside the hood. And also, now I'm going to cut the nerve which is inside the cobalt chloride. In this case, we don't have to remove the dome, because if you remove the dome, it might the cobalt chloride spill into the inside the prep, then it will make all mess up on everything. Then in this case, we have a nerve goes into the well, and we just can cut the nerve outside the well on the plastic platform. Then just cut the nerve and don't pull it. When you cut it, then we can take out the our prep and put it inside the saline. Then we add ammonium sulfide. The ammonium sulfide will react with the cobalt chloride. Then in this case, if the cobalt chloride is inside the nervous, then we see that the whole PD organ nerves will get black. Then I cut the nerve. Now I transferred our prep into the saline. Then after that, I will add a drop of ammonium sulfide, use dropper, and also remove the lid and put it here. Then remember that we should do all this stuff inside the hood. Then just add a drop of this ammonium sulfide. And after that, shake it to disperse the ammonium sulfide inside the saline. Then after, when you add ammonium sulfide into the saline that precipitates the cobalt chloride, then we should put the prep inside different concentrations of alcohol. This is alcohol dehydration process. Then I'm going to do this now. Then first I will put a little bit of 50% of alcohol inside this, and also I put the prep inside it, and I should leave it for 10 to 15 minutes. Then we have different concentrations of alcohol: 50%, then 70, 80, 90, then 100%. Then after that, we should clear the prep. Then in this case, we use methyl salicylate, and also you have to be very careful because this one is toxic. And we put a little bit of the methyl salicylate into our prep, and it will clear everything. Then it will clear all the tissues. Then you just see those nerve cells that filled with cobalt chloride.